This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 81, recorded on January 21st, 2015. Hey everybody, I'm Vincent Racaniello, and with me is Dickson Despommier.
Hello, Vincent.
How are you, Dickson?
Very well, thanks. And yourself?
It's too early to do a podcast.
No, it's perfect.
You're like,
I like this.
Not that early, actually.
No, this feels good. So how are you, Vincent?
11
I said
a.m.
I'm fine, and how are you?
I'm well.
Good.
I'm well.
Good.
Also joining us right here in the TWiP studio, Daniel Griffin.
Hello, good to be back.
Hello, Dan.
So you
Hello,
decided
Dr.
that you would continue.
Well, it's every episode by episode. We'll see how it goes. But I'm enjoying it so far.
You can't back out now.
That's right.
As you will see from our letters, people like you. It may be the first time in your life that someone likes you.
Yes. So that may bring me back.
Just be warned that
What?
it isn't all roses and... No, that's
is,
just teasing.
We have good fans. But
we have great fans.
when they're critical, it's fine for the most part.
We have terrific fans.
Although someone said recently that he was getting bored with TWiP, so here we go. We've made a change.
Well, we've made a change.
So we had quite a few responses to our first case study last week. Let's see, we had 1, 2, 3, 4, 5, 6, 7. Isn't that good?
That's superb.
So let's see what they did. The first one I got.
Should we remind the audience first, summarize the case again, just to make sure that everybody's aware of what that was
Daniel,
all about?
I actually wrote up a little summary from last week.
Okay.
I like to come prepared.
Perfect. Oh,
Unlike Dickson.
gee, that's too bad because I thought ad-libbing was the best.
It's going to show you
Yes.
up, Dickson.
Yes. So
It already
anyway,
has.
Okay.
I made sure to start that last sentence with "so," which apparently
Yeah, please.
Dixon pointed out when he says things that I don't necessarily agree with. I usually start with "so."
Speech patterns are almost irrevocable, when we're not thinking about them. But
Now let me go ahead. Summary from last week.
Okay.
We had an asymptomatic 29-year-old Bangladeshi woman with an 8 by 5 centimeter cyst in her liver that was found incidentally when she was admitted to the hospital for influenza.
right,
This woman is originally from Bangladesh,
right,
was there helping her father with his sheep, goats, and dogs 3 years prior.
right.
All serology blood test results that were checked were negative, and the cyst is remaining unchanged based on repeat imaging.
Unchanged. Remind us how long that was.
It was 3 months. It was a 3-month interval.
3 months. So when you say it's 3 months, sometimes it's a lot, and sometimes it's not a lot, depending on what causes that cyst. Yes,
And we generated a broad differential
we did.
diagnosis.
We did.
And I was actually I will say, it's great that the listeners like me, and I will say I like the listeners because I was rather impressed with a number of the emails that we received.
So
First one was from Alan. I'm going to paraphrase them. Okay? Is that okay with you?
I don't know. Ask the doctor on call.
No, it is perfect with me.
Dear Vincent and Dickson and Daniel, I always enjoy listening to TWiP here in Kona, Hawaii. Our weather today is 79F and clear, but we have just experienced a record 25-year overnight low of 54. Freezing, as few of us have either a heater or air conditioner in our home or office. One wears their hoodie and socks to bed and work this time of
Do you know
year.
what Kona, Hawaii is?
I know, we're
famous for?
pitifully wimpy. Coffee.
Bingo. Very
I am
expensive
intrigued.
coffee.
I am intrigued by your proposed inclusion of a medical case study, as long as it does not diminish your famous narrative diversions and rabbit trails. What do you think?
Could never affect that.
My clinical training and work was in and exclusively for practice in areas of the world most challenged by access to healthcare, setting up and strengthening local primary healthcare. So my diagnostic thought process went as follows. As diagnostic tests are always at a premium if available at all in these settings, the medical history and exam became especially important, as was the clinical gestalt of elder medical practitioners. Is that you? You're not elder.
He's working on it.
The left flank pain initially made one think of UTI, urinary tract infections, but when ruled out the intermediate abdominal pain and finding of a liver cyst, next made one think of an amoebic liver abscess and a round of tinidazole, which also does not go well with alcohol. As your stated tests indicated a more fluid-filled cyst than an amoebic abscess, I would have last started considering echinococcus or hydatid disease. She had the opportunity of infection, but it should have been confirmed by the ELISA. However, some 10 to 20% of cases are false negative. I didn't recall what the findings were for the kidney or spleen, which are more left flank. Still, a few months of albendazole or mebendazole every 2 weeks might both bring symptomatic relief and narrow the diagnosis, but I may still be missing something. I appreciate you highlighting symbiotic infections that need multiple species to cause pathology when the presence of just one may often be benign. I think this understanding illuminates a number of conditions. Keep up the great work. Alan is at the University of the Nations in Kona, Hawaii.
Nice.
All right, Daniel wrote just one sentence, cystic echinococcosis. Okay. Peter wrote, hi, TWiP team here. My thoughts, cystic echinococcosis.
Right.
And he gives a bunch of links for that. And then he suggests albendazole. And then at the last sentence, I have read that some inoperable cysts are left in place. What are the advantages of surgery to remove the dead cyst? Would there be more risk of complications from surgical removal or leaving the dead cyst was left in situ? Rebecca writes, Dear TWiP doctors, I've been waiting to write for some time, but the recent new design of the podcast really gave me the motivation. I was already happy to see the new episode since TWiP is my favorite of the Twix series. But the explanation of the new design really got me excited. I know I will not know all the answers since I see very few parasites working in a small hospital in Michigan. The question really got me searching and thus learning more than I would on my own. And then she tells a little story which is fun. Before I reveal my answer to the case study, I would like to share a story of my own. This was a specimen submitted on Halloween night 2014. A woman was concerned that she had passed a large worm in her stool, so she came into our ER with a jar of stool. The staff brought me the jar. My thought was that I would sort through it and try to pick out the worm. I noticed there was a large ribbon-like object in the specimen, so it turns out it was a paper towel, and then she realized it was a joke played on her by the ER staff, who had taken some soup, some beef stew, and potatoes and parsley. I don't want to have ever stew again now after that. Okay, now for her answer.
That was Mulligan's stew.
She thought it was echinococcosis. Well, I was a bit thrown because of the serological test being negative, but after looking into it further, I saw that the sensitivity of the test was around 80%, so it's possible it's a false negative. Also read that the liver is the most common organ involved. Even if I'm wrong, I had a lot of fun trying to work this case out. Thank you so much for this podcast. So Rebecca is at the Peacock Health Services in Michigan, Hastings, Michigan. Damon,
Pennock.
Yes, sir.
You said Peacock.
You have to speak into the microphone.
Pennock, not Peacock.
If you don't, we can't hear you.
I just corrected
Yeah,
that.
Pennock, it's fine. Now I have to go back and edit this, which is a lot of work for me.
Whatever.
It's a lot of work. Because I just like to
I
pop
would leave
them
it
out,
in,
you know.
actually. It sounds like
I
fun.
want people to know that you're pain.
And that he's still learning how to use a microphone.
No, that I can still read.
You got to keep your face on the mic. It's called On Mic.
Yes, sir.
Damon writes, you guys are great. I thought you wanted to hear that.
Always.
I've listened since the beginning and enjoy all the information you provide. It has only been a mere 32 years or so since my last biology class, high school, and I'm surprised how much basic information I retained. So his idea about this, I really perked up when I heard she helped her father with his sheep. Then I reviewed TWiP 7 tapeworms and decided she'd gotten dog tapeworms from her father's sheep. Okay, that's his guess. Damon, Justin. Since I used to work in a clinical animal laboratory doing hematology and urinalysis, I'd say that this still sounds like it could be a helminth, although maybe I should stop presuming all developing areas have worm issues. Treat with benzimidazoles. Robin said CE, which I guess is cystic echinococcosis, right? And he gives a long list of how to treat it and do surgery for it. Daniel, Robin is a retired ER doc who follows all of our podcasts. And so he is very, he participates quite a bit. All right, so everybody said CE. basically. What do you think of those thoughts, Daniel?
As I said, I was rather impressed with the sophistication of our listeners. And my goal, right, is to present cases that you can get. These are not going to be esoteric. Oh, I can't think of that, and then people are going to be frustrated. If you read an Agatha Christie and you pay attention, you can often figure it out.
That's right.
Some of the modern mystery writers, you're like, what?
Exactly
There's no way to get it, and I want people to be able to get these. So I'll have to say that at this point, the clinicians involved in the care of this young woman came to the same conclusions as our listeners.
Right.
So really, we don't need clinicians. We really just need to put cases up on TWiP, and then the listeners will tell us what to do. And actually, so at this point, we went ahead, and despite the negative serology, we put this woman on albendazole for 4 weeks.
Right.
Why did we do that? We did that because the history was compelling. The serology is not 100% sensitive. And as our readers quote, about 20% of the time the serology will be negative despite the presence of the disease. And part of it is that if this patient has a echinococcal disease, an early simple cyst, 20, maybe even 40% will be negative. As the disease advances, the test becomes more sensitive. But we don't want to let it advance, and we're going to discuss why. This woman gets put on albendazole for 4 weeks, and then with the fear that this is a hydatid cyst, she goes to the OR. And what do? We talked about this a little bit off mic, the three of us last time, is you have this cyst that's fairly close to the surface. Normally, if we were going to drain a cyst that we were worried was bacterial, we would go through the smallest amount of liver tissue possible. But with our concern of a hydatid cyst, we want to go through the thick of the liver so that we don't risk potentially spilling the contents of a hydatid cyst into the peritoneum.
What would that do?
That would possibly result in the patients dying on the OR table. And we've actually seen that happen a few times.
Well, anaphylaxis is what you're worried about.
That's exactly. So there's two risks. One is the immediate anaphylaxis where they die on the table. The second is that the lining of this cyst, the inside lining, is made up of cells which really are basically stem cells for the echinococcus.
You got it.
And if a single one of those cells gets out, it'll form another cyst. So there's the immediate risk of dying on the table, and then there's the risk that they'll get disease all through the abdomen.
Right.
So, to begin my sentence,
when you say
the needle goes through the thick of the liver into this cyst. The contents are drawn out. So we puncture. That's going to be the P of pair, which we will discuss. The cyst is punctured, aspirated, the A. We aspirate the fluid.
So that means it's fluid filled. It's not
so it is
solid.
fluid filled.
It is fluid filled.
The fluid is pulled out, then immediately examined under a microscope.
Right.
How experienced are the people looking through the microscopes in an OR of most hospitals in the U.S.? Not very experienced.
That's right.
When they look, they see nothing.
Right.
They don't see it. So they are left again at this point.
It's acellular.
There is material in this fluid, but they cannot see any parasitic components. They
No
can't,
potatoes or carrots.
or chunks of beef.
That's right. Dinty beef stew it's not.
But we have an experienced surgeon, a clever, concerned, careful surgeon. After aspirating, even though the initial microscopy is negative, again, the story is so compelling. He instills hypertonic saline.
That's essential.
So that if this is a hydatid cyst, that hypertonic saline will disrupt and kill all of the parasitic components.
Exactly.
Including those potential stem cells.
Exactly.
He had this planned ahead of time, I presume, right?
Yes, but actually, I've heard this operation performed so that as you aspirate fluid from the cyst, you inject an equal amount of hypertonic saline at the same time. So the cyst size never changes.
Yeah.
And it's completely replaced with this
and you leave
solution.
you leave the saline in, you don't withdraw that.
Well, another approach would be to use alcohol.
Isn't that going to mess up your liver?
Not well.
Well, they have shifted, and that is true. This technique of pair, puncture, aspiration,
Replace fluid
inspiration,
replacement.
reaspiration,
PAIR.
PAIR, puncture, aspiration, injection, reaspiration, was initially introduced in Tunisia, and there are different things that can be instilled that could kill the parasite. Hypertonic saline is probably the safest and the most common. And there are certain centers that do a lot of this, such as down in Texas, where they have a pair apparatus. It goes in and you're putting stuff in, you're taking stuff out at the same time.
Right.
Now, the surgeon then, once this has all been sterilized or so, does actually remove the cyst.
Right.
Not that is necessary, but it does get removed. I am actually in the parasitology lab and get a chance to look at the contents of this cyst. So it's a wonderful all coming together.
What color was this cyst once it was removed?
The cyst itself?
Yes.
Why do you ask? I'm curious.
Because I it's a leading question. I have an answer for it, but first I need your answer.
I was not in the ER. I was in the parasitology lab. I'm going to review the fluid.
But you never got to see the actual cyst.
I didn't get to see it this time. But the first time I saw hydatid cyst removed, I was wearing little slippers, and it was manual bagging of a patient, and I was in the third world. I was in Buer Hospital.
Right.
And the cysts were actually fairly thin. They were a whitish color.
A whitish color is perfect description. So automatically you know that we already knew from the consistency of the fluid that it's not entamoeba histolytica. It's not an amoebic cyst. So this is a fluid filled. Hydatid means fluid filled in Greek. So you've got your diagnosis half over. If the color of the cyst were white, this is another diagnostic milepost that you've crossed that says, now I have two things that tells me this is hydatid, aside from the epidemiology. But I know what you're going to say because I can look over your shoulder now and I can see the microscopic diagnostic telltale.
Yes, Dickson is looking over my shoulder at a picture I have of...
And what did you see when you looked through the microscope?
Hooklets.
There you go.
And once you find your first hooklet, it becomes easy to find more. And what are the hooklets, Dickson?
Well, now we're into the biology of this thing again. So these little protoscolices, if this were the liver of a sheep, for instance, and if the rancher or the sheep shepherd, I guess you'd call them, if they ran across this cyst upon slaughtering these animals, in traditional farming methods, they used to just take these diseased livers and feed them to the dogs because they were worthless. Why waste it? Give it to the dogs, right? Well, each one of these little protoscolices turns out to be a potential adult dog tapeworm. And they have hooklets that around the rostellum of the tapeworm itself, which is the head of the tapeworm. The whole thing is known as a scolex, but the part that holds the hooklets and the suckers, the rostellum is the hooklet part. And then the suckers are arranged around the scolex itself. And it uses these to hold onto its host with. So when you see these little hooklets, it's absolutely diagnostic for Echinococcus granulosus. Absolutely perfect. There's nothing else that produces a hooklet that shape. But they're incredibly small, so they're hard to find. And we used to have an exercise here at our parasitic disease laboratory with our second-year medical students, and we asked them to find them. And, of course, very few were able to do that because it requires a great skill on the part of the microscopist to know what they're looking for first. And we would always sit down and help them find these things. So it became a real interesting exercise. There's a whole bunch of other things I could say about this, but I think we're probably giving too many details on this. So you're going to come up with the answer for this case now.
So yes, we
made the diagnosis.
We have the diagnosis. This was, as our listeners figured out,
Bingo.
a hydatid cyst. This is Echinococcus.
So these hooklets, if they got out of the cyst, would not do anything?
They're inert. These are the they're a composite of some deposits made by the tapeworm itself, but they're acellular. So these are not the things that would spread and make a new cyst.
So she was in no danger of disseminating.
Well, I would say think of the hooklets as the teeth of the worm, right?
Correct.
There's a head of a worm. It has these hooklets. The hooklets allow it to grab. So we're seeing the teeth.
You are.
And we know that these are the teeth of an echinococcus.
That's right.
So the teeth won't get you infected, right?
So albendazole might have killed off the protoscolices in the meantime, and the only thing that's remaining of the protoscolics is the hooklets itself. So then you had the only remnant that was diagnostic.
Yes. The question would be, and I think this was brought up last time, why didn't we just leave this poor lady alone? Let me just leave that there. Why are we doing this to this poor woman? She's fine.
That's
if
true,
you do nothing,
right.
Is there a mortality to untreated echinococcus? And the answer is yes.
You betcha.
And it is about 2 to 4%.
Right.
If you leave it, this cyst will go through a period of stages over about 10 years. It starts with a simple cyst. It then can actually develop eventually, if you can get through your 10 years, to a calcified, inert, not something you need to worry about.
Right.
These are given stages CE1 through CE2 and 3. And now, if she has a simple stage 1 cyst, you can go in and do what we did. Let's say her young son, I have a young son who loves to run over and give you a huge hug, and the head's right into the belly, and often they knock the wind. I'm very excited to see me.
Right.
Playing a sport, any kind of trauma could rupture one of these cysts. And as we talked about, if you rupture this cyst, they could die immediately of anaphylaxis, or this disease could spread through the whole belly.
Exactly.
There is a risk.
Yeah.
So you do want to actually treat these people if you find them at an early
So
stage.
there was a rash, no pun intended, of deaths in Chile and Argentina among young soccer players who grew up on farms. And it's still endemic down there, by the way. So Echinococcus granulosus is still prevalent throughout the cone region of South America. And every now and then they would get kicked accidentally, or maybe on purpose in some cases, in the stomach, rupturing the cyst, and they would die on the playing field. And so this became known as a sudden death syndrome in South America, and traceable right back to the anaphylactic reaction.
How many of these have you seen in your career?
I'd say probably less than a dozen. It's really a disease that gets brought to this country, except for down in the Southwest. I've actually seen
That's right.
the New Mexico, Arizona area. Unfortunately, there still are shepherding practices where the sheep are transmitting it to the dogs, and this cycle gets set up.
So the CDC used to have a program. Peter Shantz used to be in charge of this program at CDC because he was a tapeworm expert, to go down into the American Southwest and into California as well, southern part of California, and treat the dogs for the tapeworm, the adult tapeworm, by giving them praziquantel. And this was a routine that was practiced for many, many years, and I'm not sure if they're still doing that or not.
This lady had acquired this in Bangladesh, right?
Yes.
And in Bangladesh, some people estimate 10% of the population may have this. Bangladesh is a pretty tough, swampy area
Absolutely.
to live in.
So if you work there, you might see a lot more of this.
Sure. So if you go around the world, New Zealand, Australia, England, Ireland, wherever you have sheep shepherding in traditional methods, and a lot of those are traditional farms, you've still got this infection. And in fact, they used to have it in Iceland. And so how do you think they got rid of it in Iceland? It is not there any longer. It's the only place I know of where it's actually been extincted. So what they did was they
eradicated,
killed all.
Yes, my children take issue with that. I'm not sure that that's a word, extincted. But
They eradicated all of the dogs.
Dickson likes to take
They killed
liberties.
the dogs. And then
Oh, that's too bad. They culled them.
they killed,
Yes.
they killed every one of them. And then they reintroduced them through cesareanly sectioned births of puppies to make sure that they hadn't come in contact with this. And I believe that they also coupled that with a complete slaughter of all their sheep population, and then they imported sheep that were proven to be negative for hydatid. And that's how they established it. So they have very, very strict entry requirements for animal products. And so, can you guess what an animal product might be for a place like Iceland? Try fishing flies that are made from animal furs and animal feathers and stuff like this. The possibility is if you used fox fur or some other animal that could be a harbinger for this infection. So they soak all your flies in formalin. And they don't like the fact that you might have visited farmland in your fishing travels because it could reintroduce this infection, right?
Yeah.
Yes.
Cyprus had an interesting situation along these lines too, because Turkey and Greece share that island, and one of them had a very strict control program and the other one didn't. And one of the strict control programs was a goon squad of machine gun armed teenagers in trucks that would travel throughout the countryside and shoot every dog that wasn't on a leash.
Wow.
And sheepdogs, they don't herd sheep on a leash. They're free ranging.
Yeah, right.
So it was a terrible situation for a while.
No,
really
that's
bad.
true. And even in the Southwest U.S., you still have the sheepherders with the dogs.
Sure.
So no, in your story about Iceland, it's the island is key. You're on an island you can control.
Yeah, absolutely.
The U.S. has a challenge because we have this huge border with Mexico, and
exactly.
south of that border, this is still a huge disease.
Here.
I should mention for our listeners, I have a personal connection to Iceland.
Really?
When I was engaged to my wife and I wanted to marry her, her father was at that point the U.S. ambassador to Iceland. So I had to fly to Reykjavik and ask the ambassador if it would be okay for me to, well, the ambassador and his wife, it would be okay for me to marry their daughter.
How wonderful. And
Yes.
I hope that was a unanimous decision on their part, at least. Whatever they said.
Anything else on this case?
No, I think
That's
that I have a little joke at the end.
Uh-oh.
And I stole this from another infectious disease doctor down at
Go
Walter
for it.
Reed.
Go
I should
for
give him credit, but I won't. You have children and you're a dad and you're always trying to do a good job. And I try to be involved. And we try to get our children experience to lots of things. So they can milk cows, they can milk goats. We took them one time to see sheep being born.
Nice.
And this is great and all, but there's all kinds of sheep droppings on the ground, right? So my son, Barnaby, goes out and he's doing his thing, and down he goes. And he comes to me and he's upset, but he wants his mom. But I'm gonna take care of this because I'm an involved dad. Barnaby, this is my 9-year-old. Barnaby, what's wrong? And he's got sheep stuff all over his nice jacket. He's very upset. Now, I know what he's worried about, right? So I say, Barnaby, it's okay. They don't have dogs.
Right.
And that's really the key with this disease. It's the sheep and the dogs. If
And
it's
he
just
got
the
that
sheep,
joke.
no, he went to his mom. No, this is just a joke. I mean, I'm not that bad at that. But that was just to try to seal it in people's mind. It's the sheep, the dog cycle.
Got it right.
So I just want to tell everyone that Robin had the pair described very well in his
Nice.
discourse. There's a lot of material we didn't read, and they'll be in the letters, which we publish for every episode.
He did a great job of treating them with albendazole, going through pear.
No, he wrote a paper on this.
And
Answering your question.
all the others, we didn't read all of their
We
letters.
accepted that
but,
paper for publication, by the way. So we will publish that.
you can find all the details in the letters for this episode, TWiP 81.
Right.
Now we have a paper.
Very interesting.
How did you come across this paper?
Well, actually,
Dickson?
it was secondhand. The neighbor to you in the office next door, Dr. Stephen Goff,
in whose
who
lab
was a close
Daniel
friend of all of ours,
is a member.
and who
Yes.
Dan Griffin actually works with and for, sent me an email. He got a pre-publication notification that there was an article coming out in Science that chronicled a wild animal population that was half of which was treated for a particular parasite. But they were interested in dual infections, not single infections. So it turns out to be one of the more interesting parasitic disease papers I've ever read in terms of wildlife parasitology. Okay, this involves wild animals.
So that's what caught you.
That caught my attention immediately.
Or you just didn't feel like looking for a paper on your own?
No. I was desperately searching for something that would grab us. And here it is.
What's the title, Dickson?
The title of this paper is Opposite Effects of Antihelmetic Treatment on Microbial Infection at individual versus population scales. Now, that doesn't tell you what this paper is about,
Who are
actually.
the authors?
The authors are Vanessa O. Izenwa and Anna E. Joles,
Where are they from?
and they're from the University of Georgia and the College of Veterinary Medicine.
Also Oregon State University.
Oh, also Oregon State University. That is exactly true. So the essence of this paper, before we go into the details, is that they went to South Africa, to Kruger National Park, and they culled, although they didn't kill, they culled out 209 wild Cape buffalo.
I don't think culling is the right word.
Well, they darted them. They
Culling
radio
is
collared and darted 209 wild Cape
216.
buffalo.
216.
Okay, close enough. And then in half of them,
Are these big animals, Dickson?
they're huge. They're dangerous.
So
They're-
you have to dart them.
Oh boy, do you! So you need a whole team of people in trucks.
They had two authors here. They must have had help,
No,
right?
they had a lot of help. They had tons of help. They had helicopter pilots, and they had all kinds of game wardens and people like that to make sure that they weren't eaten alive by all the other animals that also occupy Kruger National Park.
African buffalo.
Yeah, that's
Cincerus kaffir.
it's an amazing.
Have you ever seen one?
Yes, I have actually in Kenya.
Big are these big animals?
They're huge.
1,000
And they-
pounds.
Oh, more. And they stink. They do. They're dirty. They're stinky. They love swampy areas. They don't take good care of themselves. And Dan, I know you've seen them because you've spent some time in Africa.
I have. They're very impressive animals. The first time I saw them, I was walking. I don't know if it was Zambia or Zimbabwe, which country I was in at the time, but we were doing this walk because I felt like that was the way to do it outside the vehicles.
But of course it
and
is.
as you approach the Cape buffalo, the males all face you and form a ring and protect the females. And these are, as Dickson points out, these are huge, magnificent animals.
Amazing.
So what are the infections that they were interested in these buffalo?
Well, they were interested in two kinds of infections. One kind of infection induces a Th2 kind of response.
Dickson, what's a Th2
Well, we're
response?
talking about helper cells here. We're talking about B helper cells.
No, not B.
Helper T cells.
T cells.
I mean, that induces antibodies through that route.
Wait, should the immunologist jump in?
Yeah, absolutely. Please.
Let him explain it.
Anytime you feel capable, go for it.
Yeah, let me try to. Hopefully, we'll find out from our listeners whether I make this simple or more complicated. This goes back quite a way. The idea of breaking the immune response into different subtypes.
How far
And-
back does it go? No, I know the answer to that one.
Oh, go ahead with that. Tell me.
That's Robert Good's work. And other people such as that. When they did the bursa fabricius in chickens, then they got a division of the immune system into Th1 and Th2
Yeah.
responses.
And the bursa, that's where the B of B cells actually comes from. It's not the
Exactly right.
bone marrow, it's
Exactly
the bursa.
right. Exactly
Because
right.
we don't have bursts.
And we don't have one of those. We still
We
call
don't
them
have a
B
bursa.
cells.
That's
We have bone marrow, though.
We do. but
True.
it's-
Some of us more than others. Now, the division goes back actually historically, where we realize that when you have a viral infection or any kind of infection that's intracellular, there's a certain type of immune response which we call a Th1.
Right.
We think of it as cellular.
Right.
And the cytokine, the chemical messenger that is the hallmark of that response, is gamma interferon.
Exactly.
There's then a type 2 response, which rather than cellular is more humoral, more of this B cell antibody focused.
Got it.
And that has IL-6 is the big
And
cytokine.
IL-5 too for the eosinophilia that follows.
So
that as
the
well.
cytokines made by Th2 cells help B cells mature into
They do.
plasma cells.
That's
Exactly.
To make antibodies.
Sure.
Exactly.
Well, that's the simple view, but of course,
And I think
become much more complicated.
it is
How
a
so,
center-
Dickson?
Well, we now know how many interleukins there are, and it's up to what, 30
Of course,
or 40 of,
of course.
them.
But there are signatures of both.
Of course,
There are also other kinds of T cells. Just to point out, remember we talked about Th17
we
cells,
did.
right?
We
Tregs,
We
so
did. We
forth.
did. And then there's a gamma delta T. Okay, so there we're looking at intestinal helminths as a skewer of Th responses towards their immunity, which is Th2,
Right.
and away from cellular immune responses, which are classically Th1.
Mm-hmm.
And so they looked at a second set of organisms, and actually just one.
What was the first organism again?
They're strongyles. They're called strongyles. It's a group of
Strongyloides?
Well, that would be one of them, but strongyles is a general term for a classification of worms that are related to strongyloides, but there are many, many, many others.
Strongyle.
Yes, it's a general term.
These are helminths.
They are nematode helminths. They're not just helminths. They're
Not
nematode.
helminth nematodes.
No, helminths is a general term for all worms. So these are nematodes.
Did we talk about these on a previous episode?
We've got lots of episodes.
No, strongyloides we did.
We did,
you know?
of course we did.
Where do they reside?
Well, the intestinal parasite load is what they were addressing with their treatment.
So these buffalo are naturally infected,
Correct.
right?
Correct.
By these. And
These were all natural infections, by the way. Everything they did on this paper involved natural infections.
And the other, the bacterial infection that you
Was
referred to.
Mycobacterium bovis.
And this is also naturally acquired. right?
Correct. And there's a high mortality rate among water buffalo with regards to Mycobacterium bovis, but there's an equivalent story in people.
Are these wild animals?
They are. There's no such thing as a domestic Cape buffalo, I don't think.
Well, you could have one in a zoo, right?
No, I don't think you could keep these in a
No.
zoo. I think they would try to break out of the zoo or kill themselves. These are vicious.
They're really vicious animals.
These are the most feared besides hippopotami.
Huh.
Nobody's afraid of lions. If you can see a pride of lions, it's usually that they've already eaten, so that they're going to leave you alone, basically. They only eat once a week, by the way.
So this study is remarkable.
This is
in
a
sense.
remarkable study for many reasons.
Okay.
So, and I didn't know this when Steve actually emailed me the reference for this article, but upon reading it, I tried to imagine myself as the veterinarian in charge of this study and designing the study in such a way that not only did they radio collar and tag 216 Cape buffalo, and not only did they drug out the worms from half of them.
Drug out. So they all had worm infections.
They all had
right?
worm.
Did they check that?
They assumed.
didn't-
Yeah, they did. They did egg counts in their
Okay.
stool to show how many worms of which species they had.
I need to ask you this question.
Please
You
do.
got you've darted a water buffalo
You did.
or whatever,
You
an
did.
African buffalo,
Yeah.
sleeping.
Yes.
How do you get feces from it?
Well, you have this.
You put a
It's
big
not
glove.
hard.
You put a glove on and you get
Put
some.
a glove on and reach up and grab some
Okay.
and put it into a big jar and then later
Because
on.
if it were a mouse, if we pick them up, they would defecate.
well,
anyway.
that's right.
But you can't pick these up by their tail.
No, you can't. Yeah, you wouldn't dare.
So they all
try.
had strongylile infections.
Strongyle infections
Strongyle.
of various species. And in addition, they either did or did not have Mycobacterium.
No, they were all.
bovis.
No, at the beginning
And
of the
it
study, they
turned
were
out-
all. Just a minute. They were all MTB free at the beginning of the
Correct.
study.
That's what I wanted to end up saying.
At the
That is
beginning,
exactly right.
they had
so
no-
They're all infected with worms,
Right.
but none of them have tuberculosis.
Okay. So then, what did they do?
Well, they drugged up half of them for. They gave them a general antihelmetic.
Let me ask you a question.
Please, just one.
They one of many. They captured these 216 females. By the way, I don't know why. I guess the males don't show up.
That is another interesting question. but-
Anyway, over a 4-year period.
yes.
So let's say today is Thursday. Oh, look, we got one. And you decide what treatment group to put it in right there on the spot because you can't go back and
No, you can't.
round them up later. And
But
right.
But what they did was they radio collared them so they knew where they were, and they darted them every 6 months and redrugged.
But they
them.
would. They redrugged them.
Every 6 months they
Wow.
gave them antihelmetics to make sure that their parasite loads were low, and then they monitored them for
This
tuberculosis.
is amazing. So they knew
It
each
was a
individual
fabulous study.
animal was.
But look at how much work that is. I mean, for 4 years they did this, so
Yeah.
that's 8 times they went to Africa.
But
Eight
this
times-
is fun work, right?
this must be. They must have been so exhausted at the end of this study that they were. I'm sure they needed to go back home just to relax.
So that half of them got an anti-helminth, right?
Right, a general antihelmintic treatment
Drug
to get
and-
rid of all the strongyles. Right, so they kept the worm loads low. That's basically what they ended up doing, and then they measured whether they caught tuberculosis or not. Okay.
How would you do that?
Well, you can do lots of tests for this.
Sputum?
Yes, you can do that.
No, this is a great. How do you test a Cape buffalo to see if it's been infected with?
Ask him to cough into this
I,
jar.
and I should make the distinction. We keep saying MTB. I didn't say MTB. It's BTB. This is bovine
Bovine,
tuberculosis,
that's right, bovine.
as opposed to the human
Correct.
Mycobacterium tuberculosis.
So why don't you tell us about the human first?
Yeah.
Now, in humans,
Good.
maybe some of our listeners have undergone this. They take a little, those healthcare professionals, often take a little needle and they stick it into our forearm, go barely under the surface. This is an intradermal shot, and it goes way back.
What are they injecting?
So they are actually injecting purified protein from a tuberculosis culture.
Derivative. You forgot to say derivative.
PPD. The D stands for derivative.
Exactly.
And then 48 to 72 hours later, they're measuring not the redness, but the induration, the hardness that develops.
How about that?
What they are checking for is our memory Th1 response.
So what are those 4 signs you're looking for with regards to inflammation?
Well, in this case, I don't want to list them because the only thing that people should remember, it's only the tumor. The induration, the bump that develops.
Is that also true for the time test that you would do for the same?
The time test, right?
Well, it's similar.
No, it's not the time
it's similar.
test.
The history goes back to our friend Robert Koch that we talked about last time.
How about that?
And when you could finally grow this thing up, this tuberculosis, the original, think of it as a homeopathy. Why don't we take people with it and give them injections of a little bit of this, and it will make them better? Well, that didn't work. But an observant pediatrician noticed that the people that were infected would have a really strong reaction.
Right.
He then started injecting, and later managed to standardize this, and it became a way. If you have a memory of having seen tuberculosis, and not just MTB, but any of the other strains, an injection of this under the skin will give you a pretty significant reaction, and there's cutoffs based upon this. Now, are we going to do that to our Cape Buffalo? Or are we going to send Dickson out to shave a little patch?
I don't think so.
Put it under the skin?
Before you go ahead, tine test is the modern equivalent. The tines go intradermally and put a little antigen in, right?
The tine test has been replaced mainly with the PPD.
Intradermal injection, really?
Yes. And the tine test was sort of a quick and dirty.
Yeah.
It could only go in so far. It gave you intradermal. Maybe not as reliable as PPD.
And you were looking for a wheel, weren't you?
Yeah, swelling, right?
In that case, it's swelling is what we're really after.
And
there have been presentations on how do you measure the swelling.
Rubor, Calor, tumor, dolor. He got the tumor right. But it would seem to me, from my understanding, an intradermal is not so easy to do.
True.
But a tine would be a lot quicker for
it was
a Quest, but it's replaced.
True. It's been replaced.
I didn't know that.
And I'll even say that the PPD is being replaced. And in this case, we're going to use that replacement because it does not make a lot of sense to catch a big Cape Buffalo, drug him, right? If you're going to shave a little area and put this in, you're going to have to drug these guys. They're not going to let you do that.
And then a couple of days later, they're going to look at it.
Yeah, then you have to find them.
I don't think so.
So how did they look for BTB in these animals?
Well, this is great. Where we realize that all we're really doing on the surface of a human being or a Cape Buffalo is looking for memory T cells that have seen this infection before. Why do it on their skin? Why not draw a little blood, take those memory T cells, expose them to some of the proteins that they might remember, and then what do memory T cells secrete? Interferon gamma. Bring us back to Th1. So that's what they did. It's an interferon gamma release assay. And we're using that now in people because it's more specific. It's not something that reacts with BCG.
Why would that tell you? So they added pokeweed mitogen to these cells
as our positive control. Make sure we've got some T cells there
then
that are functional.
They would add BTB antigens as well.
So our positive control is going to be the pokeweed.
Okay.
Our negative control is going to be you do nothing, and you see what's your baseline gamma interferon, and then your actual test is going to be purified protein from BTB.
So as a side note, every medical student who comes into this school gets tested for tuberculosis because once they go on the wards and see patients, they don't know which of those patients has latent tuberculosis, and they could have been exposed to it, and then they have to get tested again. So I don't know how many times they get tested, but isn't it true that once you're tested, can you be tested again and be negative? Or does the test itself make you positive?
The PPD is now done at Columbia as a 2-step test. You do an initial test, you measure that. 2 weeks later, you do it again because sometimes that initial test can boost your memory, remind you. Now, I don't think that's a huge issue in our 20, 24-year-old medical students. It is probably more of a significant issue in an elderly population.
Right.
And by elderly, it's like above 90, I guess, now as I get older.
Right.
Because if you've had a long time to forget, if I tell you something, Dickson, well, okay, I probably need to remind you right away. But Vincent, I could ask about a week later.
Right.
But if it was 40 years ago, it may take a little prodding, and that's really what's going on.
So what I did ask, though, in addition to that,
but then you can test yearly, and the test itself will not make you positive.
So
it's a small enough amount that it's not going to create a memory.
Okay.
I have a little side story here.
Please.
So I did a TWiP a couple of months ago with a fellow from a company called Cepheid. Do you know this company?
Cepheid, yes.
Cepheid. They make Diagnostic. They make these amazing cartridges that are PCR assays for various infections. And you just add the clinical specimen, you put it in there, and then a couple hours later you get a result. You don't have to add anything. It's all within the cartridge. So they have... for South Africa, they want to know how many of the miners have tuberculosis. So they have a truck with this little machine in it, and they drive to mines every day. And the miners come out and they cough into a cup, and then they put this in, and they get a PCR result. And then this machine is wirelessly
PCR,
internet, and it sends the results to the National Health Service, and it can all be compiled. It's amazing.
Yeah.
But they are diagnosing MTB by a sputum. I guess you have to do a deep sputum expelling, right, or something.
It has to be a real sputum. You can't just spit up your saliva. You need to really
come from
get
down
some
into your lungs. Yeah.
Exactly. And the great thing about this Cepheid test, not only is it incredibly sensitive, more sensitive than a smear for the detection of tuberculosis, which is different than the early PCR-based. But this is a PCR-based test that tells you whether or not they have tuberculosis and even gives you information about drug resistance.
Yeah. Because they can do like 6 colors, and you can do not only identify, but 5. It's amazing.
So the term Cepheid means variable star, by the way. That's actually a variable star. So their test
this
varies?
company is amazing. Huh. They've got these for almost flu, RS, everything. It's very incredible. They check They check all our mail for anthrax spores using this test. Every piece of mail is checked. For every hour, they run a PCR test, and then they release a batch of mail based on the PCR test. It's amazing. So I guess they could have used this for their buffalo, but they don't make one for buffalo.
They don't cough that much.
No, I mean that is the way that the Cape buffalo are spreading the bovine tuberculosis. They are, and they even check it's not getting in the water. They're coughing it up. But you've got a darted water buffalo who's laying there on the ground.
You've got a very narrow window.
They also don't want to just know who has active disease. Yeah. You can get exposed to Mycobacterium tuberculosis, and 90% of people will go through their entire life and never get sick. They will wall it off. They'll control it. And in this study, they don't look at this. They're looking at how many Cape buffalo get infected. They're not actually looking here at how many are actively aerosolizing and spreading. If they were, what they could potentially do is, as we cough, we often swallow that.
We do.
And so in children don't make a great cough. In children, often you'll put a small plastic tube into the stomach and do a gastric aspirate, and you can actually detect it. So you could have potentially in these buffalo that are laying there on the ground, pass a tube into the stomach, do an aspirate, and then look for active disease.
Their stomach has a lot of other things going on in it.
How many stomachs does a Cape buffalo have?
Well, it's a ruminant, so I'm sure that you're going to get some interesting results. A lot of false positives, maybe.
Yeah, in this case, I'm not sure. I don't know if anyone's ever standardized that and can tell us sensitivity.
But still, look at all the work that went into just designing this experiment. It's not an experiment, basically. It's sort of an ecological study.
Well, they gave him a drug, Dickson. That's
an
experiment.
So they interfered with the infection.
It's an experiment.
It's an experiment at some level.
What are you talking about? It's an experiment.
But they're using
They have a control, and they have a treated population. So. It's a clinical trial. How about that?
The point is that they're using wild animals.
Although clinical usually means people, right?
And they're all females. That's another interesting
And why did they do that? We don't know why they did that.
Well, maybe they were a lot easier to capture.
So what was the effect of the drug treatment on the worm infections?
It lowered
the
egg
How
counts.
much, Dixon?
A lot.
Can you give me
look this up?
Go to the videotape, please.
I'll go to the videotape.
I'm giving you cues so that you can impress.
You are. I don't want to impress. I want to... After treatment, treated individuals were less likely to be infected with worms.
Okay.
Less likely to be infected with worms.
What's the number?
I'm working on this. They used an egg count as the way of estimating the worm count. And they... this is not Jeopardy. Come on, I have some time here. So the egg count estimates were the treated group. I know I'm seeing the numbers.
It's up there on the top there. It carries over. Fewer worm eggs than were controls.
Right. I mean, they really knocked the egg counts way, way down. So they didn't eliminate the infections, but they knocked them way down. And they talk about this later. It's not important to know what the number is. It's important to know that they created a big difference between...
I'm asking you.
Well, I'm trying to tell you the answer here.
All right, it's fine.
We don't know how many worms they got rid of. But
in the untreated group, there was no effect on the number of eggs.
This is exactly right.
Okay, that's important. So what happened with BTB? You got the pictures there. That tells the whole story.
They have great pictures. If people take a look at this actual article, not only do they have a picture of a Cape buffalo,
It's interesting that
and he's all muddy and smelly, just like Dickson warned us.
His ears are under his horns.
Yes,
that's right.
Yes,
That's
it's very cute, actually.
Right.
They look at the proportion that end up being BTB positive. These are, as we said, not necessarily active disease, but based upon our IGRA, our interferon gamma release assay, are positive. So they've actually been infected. And they saw that it was a pretty similar proportion that converted. But then they're going to tell us. Then they're going to tell us something very interesting, that the mortality rate was such that a treated buffalo with a lower worm burden that could focus its immune system on the Th1 response. I'm sort of putting this all together for them. But the treated animals lived 9 times as long,
or there was 9 times less chance of dying.
That would be actually the scientific, but the extrapolation was
because they only did the study for 4 years.
That's true.
So in the untreated group, they had 33 deaths from tuberculosis over the 4-year period. In the treated group, they only had 2.
Yeah, right.
And that was the real number to pay attention to in this case.
And that is huge.
That was an enormous difference.
It's huge if you want Cape buffalo to survive.
And this, on the outset, you're going to have a party afterwards, and you're going to get out a case of champagne, and you say, we finally know the answer. But, there's a but. There's a big but. The animals that lived, that still they had more time to become infected because they didn't die as the others were at that rate.
Yeah.
They acquired an infection which allowed them to become carriers. And because their longevity was extended, they could transmit this infection more often than the animals that were not treated for their intestinal infections. And that was their big goal, was to show the relationship here. Because why? They weren't particularly interested in Cape buffalo, by the way. Nor were they interested in their strongyls, nor were they interested in their tuberculosis.
Why then?
Well, because of what's going on in the public health arena with regards to the control of intestinal parasites and control programs for other diseases that don't relate to those intestinal parasites. Okay, so what they're concerned about is that humans living in rural areas or sub-rural areas, let's say next to large cities but still not organized into suburbs that you would recognize, acquire huge numbers of intestinal helminths. And at the same time, there are health worker groups that go out and immunize against things that you're familiar with, like polio and measles and smallpox. And they don't do smallpox anymore, but chickenpox. They do a whole bunch of immunizations. And what they're worried about is that the immune system is so focused on these worm infections that when you introduce these other organisms which have Th1 response genes responsible for the protection, that the vaccines will not work as well because they've got these intestinal helminths. So right back to the field they go. They are drugging out huge quantities of children for intestinal helminths, following that up with vaccinations for these other infections. Is it working? And that's what this kind of study addresses. If we have an animal population in the wild and we can study them Does this tell us something about the way we're treating people?
All right, so my understanding from what you're saying is that they're using these buffalo as a model.
That's correct.
But why don't we just do this in people?
Well, because they are doing this in people too. But I don't think children. Well, I'll back off of that statement right away. What is the mortality rate in children from Mycobacterium tuberculosis?
So the numbers I gave before, and these are old numbers, and I don't know how much I trust them, and I'll mention why. But we say that if an individual is exposed to MTB, that's the major pathogen, but not the only mycobacterium that affects people. There's a Mycobacterium africanum.
And Kansasi and lots of others.
And the bovis also can affect people.
Yep.
The teaching has always been that 90% of people will survive, wall it off, go through their entire life and not get sick. But there are subtle variations in that. One is that half of that 10% that get sick are going to do so within the first year or two. The second is when a child being under the age of 5 gets exposed and infected.
Okay.
That's why the BCG has spread. A child, and I've seen all these different manifestations. A child can get the TB meningitis. Right. The mycobacterium can go to the lining around the heart. I've seen not only that overseas, but I've even seen that here in the States.
Wow.
It can go to the bone.
So that's miliary. They would call that miliary tuberculosis?
Well, the bone would be Pott's disease. Miliary, like little millet seeds, is when it's tiny little lesions all through the lungs, through the body. There's lymphatic. So the children tend to get more disseminated, and I think we're going to see more than just a 10% if it's exposure and infection when you're young. And now, even worse, we have what horrible disease, what pandemic that actually is not over yet?
Well, we've got HIV.
And in the HIV, say, co-infected individual, the 10% goes out because very early in the course of HIV, there's an interference with the ability to control TB. And whether you're a child or an adult, one of the big factors increasing the tuberculosis problem worldwide is this co-infection with HIV.
So let me just give you an historical bon mot. Vincent, I know you know the answer to this question. What did Franklin Delano Roosevelt die from?
He had a heart attack or a stroke or something. What?
Died of polio.
He didn't die of
Complications
polio.
of polio?
No, he didn't. I knew you were.
He had polio.
He had polio, but he had a stroke. He had a stroke, right?
I'm with Vincent on this.
Okay, fine.
FDR is a case study in the natural history of untreated hypertension.
All right.
Yes.
And
he
smoked a lot, too.
He happened to have polio.
Did he?
Right. So what did his wife die from? This is really what I was going for. After he died, his wife started to do all kinds of pro bono work throughout the world.
Yeah.
And she went into the poorest of neighborhoods and developed a relationship with them, which lasted her entire life. She died here at this hospital, at Columbia Presbyterian Hospital. It was misdiagnosed. What do you suppose she died from? This I do know. She died of miliary tuberculosis, which she probably caught from contact with all of these populations that were underserved and underprivileged in areas where these are endemic infections. And
now I know you say that, Dickson, but when did she die? When did
she died quite a bit after Franklin Delano Roosevelt died.
Do we have any dates? Do we have a rough sense?
No, we have an exact date.
What was her name?
Her name was Eleanor Roosevelt.
Well, while we're Googling it,
1962.
She was an incredible woman.
She was fabulous.
The rates of tuberculosis in the U.S. at that time were huge.
Yeah.
You didn't need to go outside.
No, you didn't.
You didn't need to go outside this country. And I'm not even sure if you left this country, you would even at that point in time find higher rates.
But if you
that
see
was
her
that was the number one killer in the U.S. in the early part of the 1900s.
Every picture I've ever seen of Eleanor Roosevelt
I want
to
encourage people to continue to do work overseas and not take this as a lesson to avoid the less fortunate.
I have a question. Now, would having a helminth infection increase your susceptibility to HIV?
That's actually the concern that they're raising, and it does look like there is an issue when you treat the helminth infections. that does allow you to mount a better response. And clearly in the Cape Buffalo, you're improving their mortality. Now, here it's TB, but it's a very similar immune response.
Right.
And the argument that I don't think they're trying to make here is that if we treat the helminths and people live longer, well, then there'll be more people living with TB and HIV and they can spread it. No, I don't think they're making that.
No, I don't either.
Now, I do think they may be making that about the Cape Buffalo is a source of bovine tuberculosis that then spreads to all the other animals. They're kind of the core animal in this dynamic. So when you increase their life expectancy, give them more of a chance to spread, but you're not actually treating the BTB in them, you may be not only increasing the Cape Buffalo population, increasing their BTB, but you may be increasing the likelihood that spreads over to other populations.
Like scavengers.
Yes.
Hyenas and lions and that sort of thing.
So are there large programs underway to administer anti-helminth to people?
Yes. Many.
Yes, there are, and
Many.
they reference that in this this paper.
There are many.
I mean, so we have for some time wanted to treat people, right?
Absolutely.
So the idea that it will improve your ability to deal with other infections is not new, but what does this add to our goal? It doesn't really change anything, right?
No, but it gives you a heads up as to what to expect for later on down the road. I mean, if you're from a population that received a lot of antiemetic infection treatment when you're in school, and you live in a rural area where you also have a lot of malaria, for instance, and a lot of tuberculosis, as you get older, these may become problems which could surface as a carrier state for urbanization and that sort of thing.
Can these be eradicated?
Which?
These helminthic worm.
Eradication means eliminated from the planet.
Yeah.
Well, we only have one right now, which actually Dan went to a presentation on for the elimination of guinea worm.
Okay.
Which is about down to the last 100 cases or so, known cases. That doesn't say there are just 100 cases.
That's exclusively a human infection?
Yes.
So it's eradicable. Are any of these involving an animal reservoir?
No.
Or intermediate host?
No, the strongyls are direct transmission.
Okay, so they're eradicable.
They are, but I mean, you have to avoid fecal contamination, and in a Cape Buffalo, that's not going to happen.
No, in humans, not Cape Buffalo.
Oh, in humans, yeah. Okay, so the big three, we've discussed these already, are Ascaris, Trichuris, and hookworm.
Mm-hmm.
And those are all controllable in this country by just simply getting rid of the feces in a sanitary fashion, right? The majority of the world, though, and I mentioned before, about half of the world uses human feces as a fertilizer.
Right.
So if you can eliminate that practice, then you have a chance. But otherwise, no.
My final question for this minute.
Yes.
This focus on the Th2 imposed by the helminth infection, do we understand this? Is it an active mechanism by which it avoids a Th1? Is it immunosuppressive, in other words, by shifting?
Well, in some cases, yes.
Because you used the words, it focuses so much on the helminth that it can't do it. But I don't like that very much. I want to know the mechanism.
Well,
do we know?
I yield to the immunologist on my right because it's obvious that you've only got so much.
How do you know it's obvious?
Well, because they compete with each other.
I don't think it is. I think it's very complicated.
Okay.
And
it's not very obvious.
No, it's.
I'm so glad you're here. In the past, he would have just shrugged me off.
Hey, 2 against. And I feel outnumbered now.
You brought him in.
That's true. We have sown within us the seeds of our own destruction, and here's a good example of this.
No, it is very complicated. We used to like to say that with certain situations, the immune system is suppressed. I think we've moved away from that and talked about the immune system being shifted or focused. In some cases, suppressed is probably the proper word. And I think this is complicated. And I think that even though we have this article showing that when we treat the helminths, we then allow the host to have a better response to these Th1-driven pathogens. As we talked about before, even the Th1, Th2 distinction is getting old.
Sure.
And there's a simplification that helps us, but it is very complicated.
Aren't these used for immunosuppression, some of these worms, in people?
There are some trials they use I believe it's a dog hookworm in the trials, where they use it for inflammatory bowel disease.
Trichuris.
And it lasts about 3 months, and then the body clears it. The maybe the telling thing there is we don't even understand exactly how it's doing that. We talk about IL-10, we talk about different immune modulators. When we understand it fully, instead of infecting people with worms, hopefully you will take a medication.
Right.
It will you'll not have to get infected with the worm every 3 months, which some people don't like.
Well, some people don't mind it, but other people would be repulsed by the whole situation unless you did have active Crohn's disease, and then by comparison, this might seem like nothing.
Well, you can use steroid to immunosuppress some of these people.
You can,
right?
but then you've got other big problems with your immune system responses to things that you should ordinarily respond to that you don't.
Well, if you want to suppress the inflammation.
I hear what you're saying, of course.
And I don't know if, as Daniel suggested, maybe someday we can design something far more specific than a broad immunosuppressant, right?
Yeah. So I spent most of my life working on an intestinal helminth, Trichinella spiralis. It also infects muscle tissue, but the immune system is directed against the gut phase.
Is that a Th2 response?
Well, that's the idea. I mean, you'd think it would be, but there are models in mice that have no basic Th2 responses, but lots of Th1 responses. They can knock out that infection just as well, but with different mechanisms, okay? So the ultimate result was, if you had to summarize immunity to just one intestinal helminth, you would involve IL-5, IL-6, so antibody for IgE, eosinophils, plus mast cells in the gut tract, plus mucus-producing cells. So the mast cells can morph into the mucus-producing cells at the bequest of certain T-cell population. It's extremely complicated. I mean, and no one has the definitive answer because it varies with each host and the immunogenetics of each host. But somehow, each host manages to figure it out. So that means they're responding to a plethora of antigenic stimulation, which results in an imbalance at either one response or the other. But the ultimate effect is the same against the infection. So that makes it extremely
Fortunately, as we see in this paper, people are working and looking.
Right.
So
this is an intriguing result with heroic effort. I would say heroic effort. I mean, you've seen those nature shows where they've got radio communication with the helicopter, and they just went into the grove of trees, and we don't know where it went, and all of a sudden the rhino comes charging out at the, you could run into a rhino in Kruger. You could run into a pride of lions in Kruger, and you're actually not interested in them at all. You're actually after those Cape buffalo. So cobras, scorpions, all kinds of things, malaria. I mean, you've got tons of stuff going on, and in the face of all of that, they persevered and got this data, and I think it's a highly meritorious study for
This work is supported by the National Science Foundation.
Yeah, that's appropriate. That's very appropriate.
Why? Because it's ecology?
Yes, exactly right.
But it has medical implications. NSF doesn't like medical
Everything
relevant
has
stuff.
ecological implications. Everything
medical,
you do.
Medical.
Yeah, well, no.
NSF doesn't want to support work on human diseases, right?
Understood.
But this has implications for human disease.
This is right.
Also, the other interesting aspect is all the data you can find online. It's at the Dryad Digital Repository. Why are you laughing, Dickson?
I'm just amused by the fact
this
that
is a trend,
the field has advanced so far since I've gone out of it.
So they publish a graph, right?
Yeah.
You can get the data that were used to generate that
That's
graph.
fantastic.
And this is the trend. Many people are doing this. They're putting all the data online for transparency.
That's fantastic.
Now, I know that in your day, we didn't do this.
My day,
nor
we didn't
my
have
day.
the data.
We didn't have the data. All right, enough with you. Anything else we should point out here? Dickson or Daniel? Oh, we have two Ds. How about that? DDV.
That's right.
No, perhaps we want to move on to our next mystery case.
Okay.
Bingo.
Is everybody ready?
Yeah. By the way, I meant to ask, did I promise everyone who got the answer right a TWiP mug or something like that?
Well, we tried to come up with some kind of a prize on this one.
A TWiP mug? It's expensive.
Is it?
It's like $20 each.
Ooh. But the more you make, the cheaper it gets, no?
We either need a sponsor or we need some kind of drawing. We enter everyone into a drawing because my hope is we get lots and lots of correct answers.
A sponsor would be great. They could then sponsor the episode and then
have a drawing.
They could pay for the mugs, yeah.
Yeah, maybe we should have a drawing when we reach TWiP 100. And
no, well, we have to do this now, Dickson.
To the
All right, I'm going to get a sponsor for this part. Okay.
Great.
All right, next.
Here we go.
Now let me present the case. And as we go through this, I got a lot of emails sent directly to me saying, Daniel, you use a lot of medical jargon that I don't understand.
So-
Stop that. Therefore, I want to present this in a way that our physicians are excited and challenged, but also the listeners of TWiP who have become rather skilled and knowledgeable in the field of parasitism will be able to figure out.
Applied knowledge, kids.
So we start off with the we call the HPI, or the history of present illness. And this is key. This is the story. While on the infectious disease consultant service at a hospital on Long Island during April, we are asked to see a 53-year-old man who lives on the North Fork of Long Island. He told us that he had felt poorly for more than a week, with fatigue, muscle joint aches, fevers up to 102 degrees on his home thermometer, stomach upset, loose stools, and dark urine.
Goodness.
Patient was brought by his family. No one else is sick. He comes to the emergency room and is subsequently admitted, and then we take care of him while he's in the hospital. Okay, so pretty vague, right? I don't feel well. I've got a fever. I'm
Dark
achy.
urine.
No, dark urine, isn't that important?
It could be.
Bilirubin is high, so to speak.
What's the one with the Egyptian pharaoh and the dark urine?
That's a
Schistosomiasis?
very good, Vincent. Very good.
And what color?
Hematobium.
What color would the urine be?
Red.
Red.
Not just dark. It would be red.
So he would notice that it would be red.
I would think if you had blood in your urine, you'd go right to a physician.
And people do. All it takes is a drop of blood
That's right.
for a liter of urine to turn it all red, and people panic.
So
People do not like to pee red.
what is the most common false positive for peeing red that you can think of? The night before that, you had a beet salad.
Well, that's actually red stool. A lot of people
Yes.
get freaked out. I knew a physician who actually freaked out that her stool was red and she had eaten beets the day before.
But
and she
urine
didn't-
is probably
No,
a
and
good
Vincent,
indicator also.
Vincent is right. It's really more of the stool. And the stool, when it drops into the bowl, will give off this red ooze, and people are frightened. But usually not the urine.
You're thinking of asparagus urine.
No, but who looks? That's what I was going to ask, but who looks?
No,
Dixon,
people do.
When
People
you
do
blow your
look.
nose, I bet
Okay.
when you blow your nose, I bet you look in your napkin.
We all do.
Yeah, everyone
I
does.
just want to see if the worms
automatically.
came out.
Yeah. What color is that?
Exactly. Jay Leno had a whole thing about that. Okay, we'll go on.
Now let's get a little more information.
Should we not try to guess at this point because it's too early?
No, let's get a little more information.
Fine,
let's
let's go
get,
for it.
because right now it's very vague.
Right. So, do you mind if I point out some features of your dress, by the way, to the audience? Because they can't see you.
Yeah, they can't.
No, they can't. They really
Okay.
can't. So I would like Vincent
No,
to take a
no.
picture of your cufflinks, for instance.
Okay.
So they're very special cufflinks.
Oh.
These are doctor cufflinks.
Nice.
So what are they? They're the caduceus.
Yeah.
And we'll go together so we can get my wonderful bow tie today.
Exactly.
You have to smile, Dixon.
I'm smiling.
Now you close your eyes. Why do they look permanently closed?
Why do you keep harping on that?
I don't know,
I'm
Dixon.
sleeping, God's sakes. Don't wake me up.
I do harp on you. It's true.
So the tuberculosis organism is showing on his bow tie.
And my bow tie is actually an acid-fast stain.
I love
and the
it.
little red guys are
love
the
it.
MTB.
I love it. So for tuberculosis, it's a hot acid fast. And for cryptosporidium, it's a cold acid fast. Same. Without the temperature.
Interesting. I'm sure our listeners are
interesting.
excited
Did
about
you wear
that.
that-
they're jumping out of their chairs here.
Did you wear that in honor of today's paper?
I did, actually.
Yeah.
I always try to coordinate my dress with what I'm doing that day.
This
Great.
is fabulous. Next week is diarrhea. You're going to go. No, we won't. Even,
I do have a waterborne
brown-
diseases parasite.
a brown shirt.
So a little more information.
Okay.
The next thing we often move on to after we get the history of present illness is the past medical history. And this gentleman, and I'll say this is important, has a history of an acquired blood disorder. Originally I was going to name it, and I thought, no, let's just call it an acquired blood disorder that requires him to have monthly red blood cell transfusions. This has been going on for about a year now. He also has a history of hypertension, which, as we noted, is actually what killed FDR, not what Dickson said.
Well, he was immobilized by Napoleon.
Past surgical history. He had a splenectomy in childhood after trauma.
Splenectomy.
Allergies. No known adverse drug reactions that
He lives
we're
on the
aware
North
of.
Shore of Long Island?
It's the North. It's the North Fork. So this is way out east.
I know
Long
where it is.
Island, for
No, I
those
know
of you
where
in the
it
rest
is.
of the
I know
country,
where it
looks
is.
like a fish,
I know where it
except
is.
it has this tail, and there's a fork that goes
Right.
up north, a fork that goes down
All right,
south.
wheels are turning.
Okay.
Wheels are turning.
His
Plum Island.
it is not
Closer
far.
than-
It is not far from Plum Island,
right.
though.
Gardner is first.
Though I don't think he may have acquired this from Plum Island. I think not.
I don't think so.
His family history was only significant for the fact that his mother had diabetes.
Right.
Medications, he comes in on
Had
hydrochlorothiazide.
no spleen, you said.
He has no spleen. He's living on the North Fork.
That's pretty significant.
He's getting blood transfusions.
See, I'm...
Don't forget it's April. April's important.
Oh, yeah.
His medication is hydrochlorothiazide, which people refer to as a diuretic or a water pill. That's for the hypertension.
Uh-huh.
His social history, occupation, he's retired from working in a retail shop. He lives with his wife in a private home. He's never used any, as we say, toxic substances.
Right.
Not a drinker, not a smoker.
Okay.
His travel geographic history, and that'll be my little plug. Always get that. Always say, where were you born? Where
Sure,
have you
sure,
lived?
sure,
that may
sure.
help us.
Sure.
He was born in New York, and he reports only local travel.
Right.
Not a
Right.
guy who's gone far
Right.
afield.
Right.
And exposure history.
Ah.
He has 2 dogs, large mixed breeds that stay in the yard, and importantly, no sheep exposure. Okay. Then we often do what we call a review of systems. And that is where we go through, we say, anything else going on? And we ask, is your nose runny? Do your feet tingle? And in all honesty, the review of systems should get pulled back into the HPI if done properly, if we think it's relevant. And so in the future, I'm going to stop giving our listeners the review of systems. 'Cause I'm going to say, I
Yeah,
do the review of
yeah,
system and I
yeah.
put it into the history of present illness if important.
Right.
Vitals.
Yeah.
And this is something that was initiated at Bellevue. At some point, people said, vitals, who needs those? What a waste of time. Blood
Vittles,
pressure,
those are vittles, not vitals.
heart rate, temperature, who needs to know? And now we it seems so obvious.
Who needs to know? That's
His
right.
temperature is 102.6. Blood pressure is 95 over 60, which is a little bit low.
Right.
His heart rate is 115. It's a little high, above 100.
Right.
His respiratory rate is in the mid-teens. So his breathing looks pretty comfortable.
What's a CBC?
We're not, you know what we usually do? What we
Sorry,
usually
sorry.
do in medicine is before we stick a needle in somebody, we often examine them.
Really?
Yes.
So you're not going to palpate
Yes.
him for an enlarged spleen, that's for sure.
Yes, yeah, he has no spleen.
That's right. He's asplenic.
And on our physical exam, we find not much exciting.
No liver enlargement.
He does that's a good one. He has no liver enlargement. He doesn't have a spleen enlargement because the spleen's absent. He's breathing comfortably, but his heart's racing along.
His heart is a tachycardia.
Yes, he
He
has
didn't notice any.
tachycardia.
He hasn't had a recent tick infection.
Let's
No,
save
that's no. Actually, that's a good question because that would be in our exposure history.
Well, I was
and
a-
he did not report. Here we are, April.
Sure.
When did the ticks come out? Are there ticks in April? Do we have to ask in April? He could have been bitten by ticks back
Is
last
he a gardener?
summer.
Is he a gardener? Does he do
No,
that?
he doesn't. He doesn't do much outdoors. He sort of just stays in the yard, stays in the
Stays
house.
in the yard. Stays in the yard. That's important.
Yep.
He doesn't just stay in the house. stays
Could he
in the
could he
yard.
have been bitten by a tick last summer? Perhaps, but not. He doesn't report a tick exposure.
Right, but sometimes those are very difficult.
And how big are, I mean, the ticks that we worry about are the size of a poppy seed.
Correct.
When I do lectures I often bring in a couple bagels, and one has a sesame seed, and you can see that poppy seeds are tiny.
They get
They're
stuck in
tiny.
between your teeth, and they make you look.
They
That's
do.
right.
and the ticks also like to go to private places.
They do.
They like to. They're very hard to find. So
They
you,
are.
you don't always get one. I think on a board question there was, and he reports a tick exposure. But this gentleman, we don't know. He
Right.
doesn't bring it up
I mean,
specifically.
not to my
We
knowledge.
do ask because it's important in the North Fork of Long Island.
it is.
Some of the things people might think about.
That's right.
Now we do some labs. We actually,
Hey.
now that we've examined him,
Good.
we
Now you're gonna
send
draw
in
some blood.
We say, well, the phlebotomist will draw the blood.
Okay,
They actually know what they're doing.
Sorry about that. They can hit the vein rather than the artery.
And the labs are remarkable for his hematocrit
One would
being
think.
low. His red cells are a little low.
He's
His platelets are a little low.
What's the name for that?
The low platelets.
What's the name?
Thrombocytopenia.
Thrombocytopenia.
So he has thrombocytopenia and anemia. We're using big words now.
Lower medical.
Oh, and his white count is a little bit down.
Yeah.
Leukopenia.
Any shift?
No. But we'll look at a smear later and see for ourselves what the cells are looking like.
Okay.
His bilirubin is a little bit high,
It should be. It's in his urine.
right? And his liver enzymes are a little bit elevated. Not knock your socks up. They're a little elevated. The bilirubin is a little elevated.
Right.
So let's put this case together before people start asking me questions.
Right.
So we have a man in his 50s
We do.
with fever,
Yes.
generalized symptoms.
Splenectomy.
Had a splenectomy. He's got anemia. He's
Yep.
got a little bit of elevation of his liver tests. He's transfusion dependent,
Yes.
and he's from Eastern Long Island.
So transfusion dependent is an inherited disorder or a consequence of the splenectomy?
Now, he acquired a blood disorder as an adult.
Like aplastic anemia or something of this sort?
Well, it's actually not aplastic. I wasn't going to name it because I didn't think it was
Okay.
particularly
He had a
important for the
diagnosis,
listeners.
though.
He has a failure to make enough red cells. He has a
His
disorder
bone
affecting his bone marrow.
slowing down.
Sure.
It's not as active as it should be.
Yes.
Okay. What do you want us to do? Start guessing?
So at this
No, you
point,
have to guess.
so at this point, we usually try to generate what we call a differential
But
diagnosis.
Of course. So infection or not infection first? Is it a
True.
toxic reaction maybe or something like this? Or is he infected with something?
And when we see this person, we don't know we're going to be on TWiP. So our list of possibilities, and that's what a differential diagnosis, list of possibilities.
Gotta be inclusive.
And it doesn't, and I think that's key. It does need to be inclusive because if you say, you know what, there's a 5% chance this guy's, if you take care of
Put it in
20
there.
patients and you didn't consider, you're going to be wrong that 20th time, and,
That's exactly
and you're not
right.
going to get to keep practicing medicine.
That's
so
right. At least not there.
You always need to consider you start with the most likely, you go all
Right.
the way down,
Right.
you consider them all, and you manage them with this context of possibilities. Because if
Right.
you don't consider it, it's very hard to diagnose something you're not
This
considering.
is very true. So, of the non-infectious disorders in people that age, from that area, there must be a profile for this.
There are, but let us focus on the infectious.
Fine.
Otherwise, we're going to have a...
Because that's what we're doing
Yeah,
here.
we are doing infections.
All right.
We sort of know ahead of time. It's probably going to be parasitic, but initially when we see them, we don't know that. So what would be a list of things, Dickson, that you might think about?
Well, if this man hasn't left Long Island at all, and I can only think of several major infections that would possibly fall into this category. One would be a bacterial infection,
Okay.
Lyme disease.
Okay.
I would think of Lyme disease because the North Shore of Long Island has tons of deer. So Lyme disease would be a definite possibility. But there's another disease, which is a eukaryotic parasite, which is transmitted by the bites of ticks as well,
Okay.
namely babesiosis.
Okay.
So I'd think of babesiosis also. And in a person without a spleen, that's a very serious infection indeed.
Would you put that near the top of your list, down at the
It would
bottom?
be at the top of my list
Okay.
because that's the way I think. But I think babesia is an infection which rose to the top of everybody's list when they found out that on, I believe it was Block Island, or I'm blocking on the name of the other island. It was close to Block
Martha's
Island.
Vineyard?
No, it wasn't Martha's Vineyard. It was-
Nantucket?
Nantucket, that was it. And Nantucket, they actually had to post signs for Lyme disease with regards to, and babesiosis. And when they talked about babesiosis, the reason why they knew about it was because people would go there on vacation and end up dying from this infection. And these were rare people. They had been in automobile accidents or they had pneumonia as a child, and somehow they splenectomized these people. They were all splenectomized. So I remember hearing about this when it first hit the newsstand, so to speak, like HIV hit the newsstand. For Nantucket, it was babesiosis in a splenectomized person. Do not go to Nantucket because the spread of this infection there is uncontrollable, and it's a tick-borne infection. So I never forgot that, and therefore, that might be not what this is, but that certainly would be a
We'll include it.
We're gonna look
We're
for
gonna
it, of
we're gonna
course.
include that. And Nantucket is actually I went there on my honeymoon. It's a very nice place. Very
After
nice.
coming back from Iceland.
I still have a spleen, I guess, so I wasn't worried. But
Right. But yet, and when we come to the end of this, I can
So
we
we
don't
have
anecdotes.
So we have added. So we have babesiosis on
do.
our differential.
We
We have
do.
Lyme disease. Any other tick-borne diseases? Now Vincent mentioned ticks. He was
Oh,
concerned
no,
about ticks.
Rocky Mountain
Does
spotted
Vincent
fever.
Does he get to guess?
He
Do we let him
does.
guess?
He does. Please. I yield.
I was thinking Babesia as well,
Okay.
which is a tick-borne
It is.
infection.
But Lyme is a bacterial infection, right? And so is. What about the rickettsial infections?
Rocky
So
Mountain
Rocky
spotted
Mountain
fever.
spotted fever is more popular on Long Island than it is in the Rocky Mountains.
Yeah.
There are more cases described for Long Island than there are in Montana. So I'd put that down there someplace
Okay.
too. Rickettsia.
Did you do any other tests that you're willing to share?
Not yet.
No, these are just things
I want
that pop up in
us
our
to
heads.
think about what it might be. You guys are focusing on the tick-borne disease. Does it bother you that it's April? Do you get a tick-borne disease and then not manifest symptoms for how many months is this? April.
Now, this could be a smoldering infection that he caught last fall, for
Okay.
instance.
So we caught it in September and October, November,
could be,
December,
could be,
January,
could
February, March, April.
But he's been given blood.
7 months.
They
So the
give
blood
him- how
blood
often does it
transfusions
come in?
are protecting him from
Well, maybe they're diluting out the infection. How often does he get a blood transfusion?
once a month.
Yeah.
Once a month.
Every month he's
So
getting.
then we have to rule in infections that you might acquire through transfusion.
Well, the blood is checked, Dickson, right?
Yeah, you think it's checked, but
I know
there
it's
are some
checked.
things that are not being
New
checked
things,
for.
but then we wouldn't know what they were, would we?
Well, we would know some of them. Like, for instance, Chagas disease. You would know that only by the fact that it might occur. With 300,000 people in this country with Chagas
They don't
disease.
test blood for Chagas?
They do not.
Are you sure?
I am definitely sure.
Do they check blood for babesiosis?
No.
Interesting.
Do they check it for malaria?
No.
Because that's how you get transfusion malaria.
I put malaria on my list.
Okay.
Yeah.
Okay, that's fair enough. Because there have been some autochthonous cases of malaria acquired on Long Island from immigrants from areas that had relapsing varieties of malaria, namely Plasmodium vivax. So it's a remote but interesting possibility, but it's April, and mosquitoes that transmit this would not probably be out at that point.
Okay.
So I would think this is not a malaria acquisition.
Okay. Now this is
But it could be by transfusion.
now this is all the information I'm going to give our listeners.
Good.
You're welcome to.
But because you've got lab tests that will actually
Then-
show you
then
what this
we're,
is.
Then we're going to go ahead and we're going to diagnose it.
Right. But that's the short list. There could be a longer list, I guess. Blood dyscrasias that, well, I think transfusion infection. Yeah, that's, I mean, once a month. A pint of blood a month or more?
A pint. Do we use pints? Isn't that like
No.
Units.
A quart?
is that a unit?
Fill it up in a quart.
Unit.
What did the dipstick say?
It's true. We give our red blood cells in units. And
We do
not necessarily
units, but it's
I
a-
would
it's-
like a pint.
Pint is for
How
beer.
many,
Exactly.
is that a full unit or a Red Cross unit?
All right. Are we done with the case for now?
I think we're done with the case
All
for
right.
now.
You can send your
exciting.
guesses to TWiP at twiv.tv and start working. We have a few letters, Dickson.
We do.
Christian writes, I want to draw your attention to an exciting new resource for researchers studying parasitic worms. Worm-based Parasite is a new public database with genome data for more than 80 helminth species. And he gives links to the press release and to the worm-based parasite.
Right.
And this was announced by the Wellcome Trust Sanger Institute,
That's fantastic.
at which Christian is a PhD student working on parasite genomics.
Right.
So this is cool because we're just talking about worms today, Dickson.
So Christian is going to have to include another name in his next letter
Why?
because he only addressed this to you and me.
Well, this was probably received before Daniel joined
Well,
us.
exactly.
Dickson.
So we encourage him to write again.
You know, the next one is from our friend Wink, who sent the link to an article about an outbreak
that.
or a case of trichinella associated with a wild boar.
That's right.
And this is an article in Clinical Infectious Diseases. Outbreak of trichinella spiralis infection associated with a wild boar hunted at a game farm in Iowa.
Right.
You hear about this, Dickson?
I did.
April in 2011, a 50-year-old man with a history of wild boar consumption
Yep.
I guess he presented with, he went to the hospital and then they did the workup like you said, Daniel, and they found trichonellosis.
That's right.
He had harvested a wild boar from an Iowa game farm, processed the meat after returning home, and developed GI symptoms 2 days later. 4 days later, his 5 family members consumed a roast from the boar, and one additional family member was diagnosed with trichonellosis. He sought treatment 4 times after illness before being diagnosed, so they didn't get it the first 3 times.
Yeah, that's right.
They finally gave him albendazole, and then he developed atrial fibrillation after giving that. Is that typical, Daniel?
Try to figure out what was typical. The typical is being hard to diagnose. This is not a very common disease, trichinella these days. It used to be more common, but
that's right, especially in Iowa.
But and a lot of it is getting the history. Did the first doctor see him? Have you been eating undercooked boar meat lately? now if that was in the case, right?
Typical first question.
Yeah, if that was in our TWiP case. We said we have a man who comes in with eosinophilia, muscle aches, and he reports that he and his family consume
swelling in both eyes,
undercooked boar meat. It is for the hunters. Maybe we have some hunters that listen to us.
Definitely.
When you go hunting, wild game does not taste good if you overcook it.
Right.
So you are often eating your elk, your deer, your bear, your boar undercooked, relatively rare.
Correct.
What do you do if you freeze the meat for a period of time?
Be very careful here.
And how long do you have to freeze it for?
Here.
Dickson's going to jump in. How long do we have to tell us about freezing,
Well,
Dickson?
it wouldn't do you any good to freeze bear meat because the larvae will survive in a bear muscle tissue because the bear meat has an antifreeze molecule in it which prevents the muscle tissue from being damaged by ice crystal formation during hibernation. As the result, the larvae living in that same tissue is protected as well. So freezing it doesn't work for game animals. Too bad. And elk and deer wouldn't have it because they're not meat eaters.
Yes.
So you're really worried about bear, any scavenger, any meat eater in the wild. You can't get rid of it by freezing.
Wild boar.
That's right.
It wouldn't help to freeze wild boar meat.
Not.
Do they hibernate?
Wild boar may not hibernate. That's a good question. Actually, I think wild boar might be the exception because it derives from pig, obviously, and the pigs go wild, and that's what happens afterwards. So I think you could probably freeze a boar piece of tissue and kill the larvae this way. But in a bear or a cougar or another animal, like polar bears, for instance, it's useless. You can't get rid of it this way.
So I would guess that he did not recognize that trichinella is a problem with wild animals, and so we need. And they point out in this article because there's not much trichinella in this country.
Correct.
We need to educate hunters and the broader population for the potential.
There's not much trichinella in the human population, but that doesn't say what's in the animal population. And there's a ton of it in the animal population. There is.
Apparently.
They did a survey once of the bears of Yellowstone Park that died. They didn't find a single bear that wasn't infected. Now, I know that there's a connection here to Yellowstone Park because Dan's mother has been involved in some reintroductions of predators.
The wolves, actually. Yes.
The wolves. So his mother, how many guys out there could claim that their mother was responsible for introducing wolves back into Yellowstone Park? I think that's fantastic, by the way. That's the best thing you ever told me. And you've told me a lot of great things, by the way. So in this case, the wolves, by the way, would end up with a lot of trichinosis as well.
The poor wolves.
But most people don't eat wolves. They just shoot them.
Yes.
Right.
Unfortunately.
That's right. So I knew about this
Can
case.
you read the next one, Dick?
Sure, I can.
You got to read.
You got to keep the mic. You got to talk
I'm
into
going
the
to try
mic.
to do both of these things at the same time. But it's hard because chewing gum and walking, it gives me a big challenge. So this is a Robert letter that Robert writes. Sorry for replying to an old episode, but I thought you might want the following information if you haven't heard of it from anyone else yet. Fiona, who wrote in referring to rope worms, said she was using MMS enemas to treat them. Here is some added information that MMS, that stands for Miracle Mineral Supplement, refers to a handful of different chlorine dioxide evolving solutions, and the usual dosing being increasing doses until the side effects require backing down orally until you can no longer avoid emesis, or the enemas until diarrhea. The rope worms, in quotes, seem to be sloughed off intestinal mucosa. MMS has been linked to deaths both due to side effects and delay or refusal of proven treatments for various diseases. While the eponymous miracle mineral solution, I'm sorry, my computer just said it's loading, so I have to now scroll. I just lost the whole website. I just lost everything.
While the eponymous miracle mineral solution has been banned in various markets, people continue to buy the chemicals that can be used to generate chlorine dioxide and dose themselves in the hope of dealing with their various medical problems. I hope Fiona is doing better and no longer using chlorine bleach internally.
Chlorine bleach?
Basically what it is, chlorine dioxide.
Yes.
You know about this MMS?
No, fortunately not.
So this lady had written in saying she had ropeworms and
I thought chlorine was sodium hypochlorite.
She couldn't figure out what it was. Sodium hypochlorite, yeah. So I don't know why he says chlorine bleach.
I don't either.
Because this is chlorine dioxide.
Sounds horrible no matter what it is.
Anyway, thank you for your ongoing efforts to produce the This Weekend podcast. All right. We have a horn blowing outside.
We
Did you hear it?
Yeah, well, I'm not responsible for that one.
No, you know what happens?
I do a lot of bad things, but I didn't do that one.
We have a rather small street going by the medical center. It's Haven Avenue, right?
We do.
Right. And it gets blocked.
Yes.
And it sounds like a truck is pissed off and wants to get through, and he is just sitting on his horn.
Right.
Yeah, that's life. Eric writes, hello, Dickson and Vincent. I just came across this video. It shows the extraction of a bizarrely long worm from a praying mantis. It's hard to believe that the mantis was still alive with that thing in it. At first, this was just a cringe-worthy video that I wanted to share with TWiP as maybe a listener pick of the week, but then I read a bit about the parasite, which turns out to be a nematode called a horsehair worm.
A remethod.
You know that?
We did a thing on that.
We did?
Yeah, we did.
I don't remember.
We did.
It's a very interesting parasite, one of those that alters host behavior, which has been discussed several times on TWiP.
Remember the grasshoppers?
Yeah. Yeah, that's right. They jump in the water.
That's right.
And then the fish eat them, and it messes up the pond ecology.
No, there was this trout. And actually, this fosters the completion of the life cycle.
Yeah. It's a parasite of arthropods and infects beetles, cockroaches, grasshoppers, and even some crustaceans.
Yeah, well, methods.
I haven't delved very deeply into the life cycle, but I read that the adults are free living in fresh water, but somehow their eggs find their way to their arthropod hosts, and eventually infections will have an effect on the host's brain, which drives it to seek water and drown, thus releasing the adult form of the worm. That's what the crickets did, right?
Yep, and the grasshoppers.
Or grasshoppers in that study.
Yeah.
These fascinating mind-altering parasites have been discussed on TWiP in the past, but I think it would be fun to have an episode dedicated to a vast array of these bizarre and in some cases grisly examples of how parasites affect their host's behavior.
Yeah.
Another example, which I think has been mentioned in passing on a previous episode, is cordyceps. Is that the one we did? Which one did we do that infects the grasshoppers that makes them jump into the pond?
Go back and look it up.
All right. Anyways, I'd like to say that I greatly enjoy TWiP and all the other Twix podcasts, particularly TWiV, since viruses are my main area of interest.
Oh, good.
And I am always eager for the next episodes to arrive. I admit I'm one of many who would love a more frequent TWiP schedule, but I understand that you're both very busy, so I suppose monthly-ish will be enough to give me my parasite fix. Kindest regards, Eric. Now, Eric, we are doing this twice a month.
We're working on it.
This is actually a scheduled episode.
We are. This is a scheduled episode.
So he sends links to the videos. And then one quick one from Jeffrey, who says, hey, TWippers, I'm really excited about the new format and the addition of Daniel to the team. It seems like he will add a really interesting perspective to the talk and maybe some great tangential stories, which I love, by the way. Keep on twipping. Jeff is in Seattle, where it is mostly cloudy, 46F, 8C, 60% chance of precipitation, 90% humidity, and wind 0.
Right. Dreary. London.
So, Daniel, you are a hit. As he looks at his email on his phone to make sure he doesn't have
No, I was actually looking about deep freezing and killing trichinella in wild game, which I learned something from Dickson today, maybe.
Come on.
He is the world expert on trichinella, so we hope he's right.
Kids, trust me on this one. I know the answer here.
Yes.
I've heard that before, Dickson.
And when you have, it was correct.
Yes.
TWiP81. You can find it on iTunes, microbeworld.org/TWiP. And do send us your questions and comments, as well as your guesses on this week's case history, to twip@twiv.org. TV. Dickson Despommier can be found at medicalecology.com and also trichinella.org. Those are relevant to this
That's
episode.
exactly right.
Thank you,
Exactly.
Dickson.
You're quite welcome. What fun.
It is fun.
We had a good time this time.
Well, we always do.
We had a better time this time. We had 1/3 more fun.
That's right. Daniel Griffin is here at Columbia University Medical Center. Thank you, Daniel.
Thank you. It's a pleasure.
Yes, good having you. I'm Vincent Racaniello. You can find me at virology.ws. The music that you hear on TWiP is composed and performed. Who does it, Dickson?
Alan Jenkins.
No, it's not Alan Jenkins. You've heard this 80 times.
Jenkins. Well, it's Jenkins. I don't know.
Ronald Jenkins.
Ronald Jenkins. Okay, Ronald Jenkins. Okay,
And I wrote to him and asked for permission. And he said, absolutely. And he's also done music for Urban Ag. And he really thinks, and people go to his site and
Ronald.
buy some music because they heard him on our
Good
shows.
deal.
You can find his work at ronaldjenkins.com. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP
Is parasitic.